designed this OmicsLogic portal, and I want to stress these two words here: omics, so variety of omics types, and that includes phenomics, that includes genomics, transcriptomics, etc. So many different types of data, as well as the logic part of it. And what is really logic? Logic means that we don't just apply in a textbook fashion. I know how to use this method, I'm going to apply this method to a data set. The logical part of it is always asking the question why, and then asking the question how. And that is what we're going to focus on in this program. Why would we apply a method, or how would we apply a method so that we come to logical conclusions? Because ultimately the goal of analysis is to extract meaningful insights from data that are reproducible and that anyone else can go through the steps that you have taken and get the same results, right? So that's the meaning of being reproducible, and a very critical factor in this whole domain of data science and bioinformatics. So let's take a look at how these topics are going to be organized in the context of this program. So the program consists of really interactive sessions like today, and we are always looking forward to getting as much information about you and about your struggles, challenges, or questions before each session. So try to keep that in mind. And then introducing some kind of an example project or an example method, pointing you to resources where you can learn in detail and in great depth the specific method or application, and then practicing together. So a lot of what this program is about is not just the interactive sessions, but the actual materials that you will have access to, because they will fill in the gaps and provide you with the necessary depth so that you can advance in your understanding. So the first part of the program is going to be focused on having a good sense of data and preparing that data for analysis. And then in the second part of the program, we will talk about some of the methods for unsupervised and supervised methods of analysis, including how to validate and prepare your methods for production. And so there are a lot of challenges along the way, but hopefully we will be able to do that if we're able to combine these asynchronous materials that you will be able to practice through on your own, and the interactive sessions that we will hold today. And so what we will do today is kind of get started with that process, but I think it's important for us to lay out the full path so that you have a sense of where we're going, right? Where are we going to end up at the end of this process? So the projects especially will be useful for those of you that don't want to just learn about the methods, but really see how these methods are specifically used to address questions around a different number of data sets. So what kinds of data sets and projects are we going to look at? And I'm going to give you just a few examples in the next few minutes. So first of all, diagnostics and cancer biology, right? So I think a lot of people have indicated that this is an interesting topic. What are some of the factors for diagnostics in cancer biology? So recently, in the past several decades, there has been a lot of progress in understanding cancer variability among patients and specific risk factors, as well as biomarkers that can guide decisions around clinical diagnosis and treatment selection. So we're going to take a look at several examples of projects where that is a factor. The second area that we will look at is single cell and tissue composition. So as you probably can imagine, the single cell sequencing has really brought in a tremendous amount of data, but also has required an additional set of methods that we're going to learn about, and that deals with dimensionality reduction, and again, markers for specific indicators or tissues that we can understand. And then finally, we will talk about other types of data, for example, microbiome and infection. We'll talk about infectious diseases. We'll talk about how those are changing the microbiome or the environment with microorganisms, and how can we analyze such data. And a lot of this data is going to be available directly from the public domain. So briefly, just to talk about that, as we see in this pandemic, there has been a tremendous growth of the data that is available in almost real time. So today, when you go to NCBI and you start researching any kind of topic, you will see right away that for each topic, as the years progress, there's more and more information available, and that includes raw data as well as publications. And so we need to appreciate the fact that the publications really represent maybe 5% of the information that is contained in that data. And so there's a lot of available public data for us to study and mine using these different methods, but we have to do this with understanding. We really have to understand the methods and the data and match the methods that we select with the data that we would like to analyze. So for that, the first part of the program will also talk about a variety of data types. And so here we have a connection for multi-omics data analysis and integration. And I will briefly speak about this because it's really important to understand how these different data types are different from each other. So one type of data, for example, phenotypic data. Phenotypic data could be characterized in many different ways, and it could refer to raw data. For example, imaging can be classified as raw data, or clinical data, where clinical data is really the annotated raw data that is based on an opinion, based on a taxonomy, based on some kind of an understanding of whatever condition we are looking at. So that all falls under this domain of phenomics or phenotypic data. The second part that we will talk about is genomics. So what is specific and unique about genomic data is that, as you know, the DNA is comprised of just 4 different types of nucleotides. So when we look at these 4 different nucleotides, first of all, this is a very specific type of data. These nucleotides appear together, not in a random fashion, but somehow in an interconnected fashion. So the sequence part is very critical. But when we want to compare different conditions, there are multiple ways that we can interpret the data from genomic sequencing. For example, mutability with frequency of mutations, or by somatic score. How specific is this to a somatic mutation? So understanding what the data means and linking it to the right kinds of methods will be greatly helpful when you start dealing with those kinds of data. Epigenomics, when we speak about epigenomic signal, DNA methylation or whole genome bisulfite sequencing, we're really talking about specific regions that are present on the DNA, and those regions have certain characteristics. They could be methylated or unmethylated, or those could be several different types of histone modification. So understanding, again, the biology of chromatin packing, of silencing through DNA methylation, and how we can measure the impact of such epigenomic data on the actual phenotype is important here as well. And then finally, we will talk about gene and isoform expression, how we can measure the levels of expression or look at variation at the alternative splicing level and decide how do we use this information for the specific challenge that we would like to address. Okay, great. So I see a lot of people have already done this course. This is excellent that you have done this course, because today is the session where we will try to load some of these data types and work with them. But because not everybody has done this course, I do want to have this review first. And so when we get to the practical session, I am sure that you will appreciate that much more than just doing this without kind of this reminder. So for those of you that have not done any of these types of data, have not worked with any type of these data, I do want to point you to a course that you are welcome to take a look at. And that course is called Bytes and Molecules. So if you go to this tab called Courses, you can type in here bytes and molecules. And this course will provide you with the terminology and examples of how this data is generated and what does it represent. So make sure that you look at these lessons, and here you have a button called Load More. and you can add here some applications of this data, including an introduction to bioinformatics languages. So if you haven't done any of this background training, please take a look at some of these lessons here in the Bytes and Molecules course. Okay, so what do we do with this data, right? So again, we have these different types of data, different omics type of data. We have to understand how this data is generated. And ultimately, what we have to do with the raw data sets is we have to process this data to make it structured, right? And so we'll spend just a little bit of time speaking about what structured and unstructured data look like. And that will be done in our next session that will be dedicated to the different types of omics data. And then we are going to try and analyze some of this data using our big data analysis platform. And so what's important here is to really understand the link between stepwise processing of data. So these are individual steps, they're algorithms. We need to understand the settings of these algorithms, and we need to understand what do these algorithms actually do. Once you understand that, it will provide a much better understanding of the final output, right? And you will be able to answer deep questions about the results of your analysis, linking it back to where the data came from and how it was prepared, right? And there are a lot of challenges, by the way, related to finding data online that somebody else processed and not really having a good grasp of how it was prepared for analysis before you do the downstream and exciting, so to say, visualization and biological interpretation. So we do have to address that in detail as well. And ultimately, once we have the structured data, we're also going to be going to this aspect of integration, because ultimately biology, what is specific about biology is that it is a complex interaction at multiple levels that we really have to appreciate. When we take a gene, an isoform, a peptide, or we take a look at interaction between different proteins, it is the collection of information around that, the context, that really plays a role in our ability to make reliable and valid interpretation of what we are seeing. So what we're going to try and do is very quickly go through different examples and placing them back into the context of where that biological system helps us understand the interaction and the significance of specific aspects of what we find during the analysis stage. And so here you can see from sequencing to comparing different conditions to trying to interpret protein-protein interactions, or maybe understanding the taxonomy or the organization of the different types of organisms that we have to really evaluate some signal that we find in a statistical or a machine learning approach, right? Which is just dealing with numeric data. So what we will try to do is take that biological context and then take a look at this process of reproducible statistical analysis in order to find inference, right? To make some kind of an understanding that is generalizable about this biological condition in general, and not only focus on a specific experiment and what that experiment kind of tells us, right? Because we do have to generalize. Okay, so let me just pause here 1 second. I see several questions, so let me address those questions. Is it possible that there will be some exercise related to immunoinformatics? Okay, so definitely possible. Again, that is the reason why we asked you to fill out the survey. Whatever you put in the survey, if you are interested in immunoinformatics and you want to do PCR repertoires, right? Or you want to look at, some kind of epitope mapping or whatever it is that you are interested in, that is what we ask you to put in that form that helps us to learn more about your background and your interests. Indrani is asking, definitely you can find a number of online quizzes on each of the fundamentals. Yes, a lot of the fundamentals are covered. We have some coursework on fundamentals. You can also find a lot of online materials on a lot of these topics, right? And so the goal of this program is to connect those together into some kind of a meaningful sequence of sessions that hopefully provides you with a broad view of methodology and a specific focus on the data science aspect of all of these together. So as we spoke about different omics data, let us briefly talk about some of this challenge with structured and unstructured data. So in reality, it is much easier to work with structured data, but it is not as available as unstructured data. In fact, in 2020, over 60% of available data was unstructured. And that is why it's important to understand that most of these data sets come in an unstructured way. And once we have access to the unstructured data, we should be aware of the steps to make it structured. And then we should be able to understand how do we actually work with that structured data in the context of a technical environment that allows us to perform analysis and get some results. And so we'll talk about today how to organize our data into different objects, vectors, data frames, matrices, how to load them, how to explore them, how to visualize them, etc. We will also look at the significance of understanding data properties from a statistical perspective. So a lot of the methods that we will cover in this program have assumptions about data. These assumptions might be about distribution, about the mean. They could be reliant on different normalization techniques or transformation techniques. And so we need to be able to understand, what do these techniques tell us about the data, and how do we find the appropriate methods for preparing the data for analysis. We will also talk about how this understanding could be a part of your exploratory process, so that the thresholds and the settings that you use in all of these steps have a meaning, right? You know how to answer the question why. Why did you choose a threshold of 5 or 1,000? How did you identify this kind of a pattern or that kind of a pattern? What does it actually speak to, both from a biological and a data point of view? And then we will turn that understanding into a contrast, right? So you should be able to look at the way your data is organized and then make decisions about what to do next. And a critical part of that is exploration, but also it's designing the contrasts, the thresholds, and the settings that you should be using. And so we hope that we will address this in the next couple of sessions. So after we do that, we're actually going to take a look at a variety of methods. Now, in data science, I think the most exciting part is that we have easy to use methods of data mining, machine learning, and deep learning. And when I say easy to use, I mean literally one line of code or maximum several lines of code to really perform complex analysis of data. And because it is so easy to use these methods, it is also very important to understand, what do these methods actually do? How do they differ from each other? What are the specific settings that they have? And what are the assumptions that they have about the data, so that the results are interpretable? So that will be a large portion of this program, and we will cover methods that are unsupervised, where we do clustering or we do dimensionality reduction. And we'll also talk about supervised methods of analysis. So supervised methods of analysis like classification and regression, but also we will talk about how we can use them in a variety of other ways, like using them for dimensionality reduction to study features that they extract from the data, or understanding feature significance and feature selection. How do we actually use those to understand a lot more about the data and be able to interpret them? Okay, so the next thing that we want to do in today's session, and again, let me know if you do have any questions. If you have any questions, I can answer those. But if you don't have any more questions at this point, I will pass this on to Dr. Raghav, who will kind of show you technically and practically how to get started with some of these exercises. Now, before I pass it on to him, I see a question. I am interested in using NLP methods for data text mining. Does that fit into the methods mentioned here? So we won't directly speak about NLP from, let's say, publications or something like that. Some of the methods that you will find here are related, but if you're only interested in NLP specifically and you have that in mind, this program is probably not going to be the right fit for you. I do recommend the research fellowship program where you're more open to the types of methods. So that's something that we can discuss later. So if there are no other questions, I will pass it to Dr. Raghav and ask him to kind of give you some practical examples of some of these things to get started with.
Thank you very much, Elia. And if